just would like to say a few words about Dr. Slavica, our guest speaker today. Dr. Slavica Tudzarova's research is focused on the crosstalk between metabolism, DNA replication, initiation, and cell cycle in human cells and tissues. Defects in these processes commonly occur during malignant transformation or human diseases like diabetes and neurodegeneration, and involve abrogation of checkpoint control and unscheduled replication, which leads to apoptosis and cell loss. As a part of this program, she is investigating the abnormal mitochondrial morphology and function in diabetes that interferes with synthesis of beta cell replication during compensatory regeneration. Her group is now working on fostering beta cell regeneration through strategies that restore normal mitochondrial network and function, thereby preserving functional beta cells. After obtaining her Bachelor in Science in Pharmacology at the University of St. Cyril Methodius in Macedonia, Dr. Slavica Tudzarova underwent training in tumor biology at the University of Vienna, Austria, where she obtained also her PhD in 2003 at the Institute for Tumor Biology and Cancer Research. In 2006, she joined University College London in the United Kingdom, where she was postdoctoral fellow until 2013. In 2014, she established her independent research program funded by Ernst Young Stiftung and Industrial Research Collaboration before being appointed to an assistant professor at the Larry Hillblom Islet Research Center at David Griffin School of Medicine at UCLA in 2015. So please welcome Dr. Slavica and thank you.
So first of all, thank you so much for this wonderful opportunity, and I can't start other way but expressing my gratitude to Father Blaško for this thoughtfulness to put this speech and to introduce me at such a celebratory moment for all of us, celebrating the Nativity of the Most Holy Theotokos. We all came and enjoyed, and I can only say that I am more than flattered and blessed in terms also of my work, which has to continue by this opportunity. So I will immediately start. I would like to introduce you with the novel insights into the mechanisms of development of diabetes and how this might shed some light on its putative link to cancer. So let me start first with diabetes. You know that diabetes is the sugar disease, and it is manifested with uncontrolled, so higher levels of sugar in the blood. And if we can simplify the main cause that leads to diabetes, to these uncontrolled levels that we call hyperglycemia, they can come from two sources. A, aberrant insulin secretion, or loss of insulin secretion, or loss of insulin action. So insulin is a hormone that is secreted by specialized cells that are part of the so-called endocrine core of pancreas. And these beta cells undergo injury during diabetes. And interesting is that over sometimes a decade or more, these changes that precede the serious onset of diabetes go on hiatus before it happens to formation of this abnormality in the blood, which is called hyperglycemia or higher sugar levels. And once there is the glycemic control, so to control these levels seems to be a very big challenge. And sometimes this expands to various failures of various tissues and organs, and in particular affected are kidneys and heart. But interesting is that very much like in cancer, diabetes is just one terminological umbrella for many diseases, not one disease. And how this relates to cancer, cancer is similar. So now, for example, clinic tries to advance the approach to treat cancer by developing so-called precision therapy and genetically sequencing the tissue from the tumors, identifying what type or subtype of tumor exists in the patient, and then custom tailor the therapy for the patient. So that, oh, it's stopped here. I have to reload it. I think, Nikolai, can you help? So basically, as I said, it's one term, diabetes, but it actually consists of various diseases. And here we include, for example, type 1 diabetes, when there is obviously a failure of the immune system, so there is alert immune system. So can I go just quickly, right?
Yeah.
Okay, so the problems with the immune system, and type 2 diabetes, which is typically, it was actually a disease of the elderly. Then gestational diabetes that occurs during pregnancy, and some subtypes of diabetes not so well known. But interesting is one which is called secondary diabetes mellitus, or sugar disease, that actually occurs as a comorbidity, as additional disease when a patient has a cancer, and in particular associated with pancreatic cancer. So, Glasnia? Okay, yes. So one of the ideas is that these two actually world epidemics, diabetes and cancer, of a similar size, and it is expected 2020 to arise with about 500 million of people which will suffer cumulatively from prediabetes and diabetes, that one of the theory is they share same common risk factors. And one of the risk factors here depicted is called obesity, and comes through the so-called glutinous lifestyle, or one uncontrolled intake of calories in form of nutrition and alcohol, for example. And this is just one example that compares proportion of population. When proportion of population that is affected by obesity or diabetes is smaller, you see beige or yellowish color, and then it turns out to orange when it raises, and then the highest population affected, it's depicted in red. You can see it across 20 years, and what you can see through this example is that the trend in development of raising proportion of population affected by obesity is very similar to diabetes, which might lead us to conclude that they are very strongly linked. But I can tell you straightforward that the link is correlational, so it's just indirect and not direct. And the reason for that is, out of 100% of all obese people, only 1/5, only 20% will develop diabetes. Opening up the question, what actually drives diabetes? Do we really know what are the risk factors, right? So, type 2 diabetes is one of the most common types of diabetes. It accounts for 90 to 95% of all types of diabetes. And how it starts, normally starts with one interesting reaction on the, so we call them peripheral tissue. This means fat, for example, muscles and brain. When we have uncontrolled intake of nutrients, alcohol, so calories in our organism, they start to defend themselves by ignoring the action of insulin. And this is called insulin resistance. So why? First of all, insulin function is to bring the sugar as energy fuel in these tissues. But when we overwhelm the organism, interestingly, these tissues begin to defend, to ignore the action of insulin, and this is called insulin resistance. And what does it bring? Why is this so challenging for the organism? Because then the cells that are specialized to secrete insulin have more work to do. So they are challenged to hard work. And it happens to be that they can cope with this situation for some time, But in diabetes, by some point, these so-called beta cells fail. They are exhausted, and then it comes to dysregulation of secretion of insulin, so they cannot secrete insulin, and there it comes to the sincere onset of diabetes. So what are the other factors, risk factors? I told you one, obesity, but pointing out that not all of these factors are actually causal factors. Lifestyle factors are treated as more factors that create environment, they set the stage, but they are not real risk factors. And I will tell you one for comparison. For example, lifestyle factor is sedentary lifestyle, right? Or for example, less known exposure to the blue light of the smart media during the night, because this interferes with our so-called inner clock. And you will not believe how many tissues, cells and organs in our body depend on actually distinguishing between day and night. So there are other factors, but all of them have just a feeding, supporting effect in developing of diabetes. There is one factor, however, that is considered serious, and it's interestingly associated with how much mass, so mass or volume, or available beta cells we are born. So some people are born with critical bigger mass on cells producers of insulin, and it's functional. So if they're challenged with kind of negative lifestyle, they will resist longer before they get some symptoms or prediabetes. While the others that are born, unfortunately, with smaller and non-functional, they can be challenged towards development of diabetes quicker. So this means you can distinguish various factors that lead to these big epidemics. So one are lifestyle, which are more supporting, and they are real, which are present in what complement of beta cells we are born, and which will be challenged by the way we live and our behavior. So ethnicity and race is a big factor. For example, Asian Americans, African Americans, Latino Americans, Hawaiians, and Pacific Islanders have high risk of development of diabetes. And interestingly, how the lifestyle can just change the history of one disease. Now we know, because type 2 diabetes was actually disease of the elderly. But nowadays, because we changed massively the lifestyle, and this became fixated even at the early age, now we have more adolescents and children suffering from type 2 diabetes. So now I just want to put you in one bigger picture how diabetes lately has been associated with cancer and why. So it happened about 10 years ago that epidemiological studies, so studies that accumulate statistical data and recruit, so acquire a relatively representative, we call cohorts, a number of patients. So these studies pointed out that diabetes is setting the stage for development of cancer. And interestingly, so there were very big studies and covered various types of cancer, and they found actually that these two diseases are linked in terms that diabetes enhances the risk of development or mortality from cancer for twofold, and this refers to all types of cancer with only one exception, where diabetes reduces the risk, and this is prostate cancer. So it is very difficult to understand, and still we are exploring what are the causes which link these two diseases. As I said, some believe that just they are the common risk factors, but it seems that expand to more. What is already very established is the link between diabetes and pancreatic cancer. And it became a rule that, for example, when higher blood sugar and diabetes appear in lean people, so not obese, but lean people, with no familiar history in diabetes at an age between 45 and 50 years, it is considered as a sufficient finding in order to screen for pancreatic cancer. Also, for example, elderly people with new onset of diabetes have 3 times higher risk of developing pancreatic cancer in 3 years than people who do not have diabetes. This was confirmed with some additional studies. But that is from all types of cancer. It seems that pancreatic cancer that coexist at the same location, pancreas, with injury of insulin-producing cells, in diabetes seems to be most intimately linked. So now, this is a little bit complex, but I will explain to you. So this diagram is collecting the ideas coming from the scientific findings, how diabetes sets the stage for development of cancer. So, as I told you, the diabetes starts with abnormal reaction, abnormal behavior of tissues like fat and muscles. They become insensitive to the action of insulin, and we call this insulin resistance. And then after that, it comes to so-called metabolic syndrome. So there are a lot of rearrangements in the metabolism of the body. And this seems to change one really important thing, and that's the way how we generate energy. So keep this in your mind, because I will refer to this later in my speech. So this all proceeds in a sequel of changes which lead first the cells that produce insulin, so-called beta cells. First they try to adapt. So when there is higher level of sugar in the blood, they basically secrete more insulin and they maintain the sugar normal in the first phase. However, this continues, the challenge continues, and because of various factors, and then at some point these beta cells fail. They are exhausted and they cannot control anymore the blood sugar, the levels of the sugar in the blood. So this is very dangerous situation, because this means that somehow now the body is challenged by out of balance, which we call homeostasis. We don't have homeostasis, we don't have balance. And the factor is very high levels of sugar, and sugar is toxic. But also it perpetuates in different changes, like for example, lipid metabolism is changed. There comes the lipid toxicity. And here the scene becomes really serious, because this toxic situation begins to affect individually the cells, to damage them, and this goes further. They also damage our genome, so our genetic information. And that is really critical point, possibly critical point to triggering cancer, because cancer cells has one interesting attitude. Very normal physiological cell in our body, when it's damaged, can undergo cell death, which is called apoptosis. It's a Greek name, and it is depicting falling off the petals of the flower. So every single normal cell, but the cancer cell ignores that it's damaged and takes the cues, various cues from the environment, to ignore that and continue to replicate, to multiply. So these cues can come from this sequel before onset of diabetes, but also from the toxicity of the stage when we have higher levels of sugar or lipids, which can affect, as I said, membranes or, for example, our genome. And this together might lead or trigger or support feed the development of cancer. Situation is not so easy, because when a person develops first pancreatic cancer, also it develops diabetes. So there is certain reverse causality between these two morbidities or diseases. But now let me tell you my story. So my story started when I moved here in Los Angeles 2 years ago, and basically I came only with cancer background. Why I shifted, this was fate determining, I would say, shift of my research here in LA, was because at one talk which I gave in Istanbul, where the two continents meet, I met a group of people which basically impressed me from the third continent, from US, from America. And they were impressed that the concepts which I discussed could be very much applicable on these insulin-producing cells in diabetes. So I will tell you now my story and my findings, and how they possibly can shed some light for further research into the link between diabetes and cancer. So what you see here is so-called human islet. Islet is a collection of beta cells that exist in the pancreas, and beta cells are cells that are specialized in secreting insulin. So here is depicted, I don't know whether you see the red, with the staining that stains the insulin. So wherever there is insulin, there are the cells which are actually in charge of secreting insulin. They are grouped with islets, and these islets, there are 1 million of them scattered throughout the pancreas. So what they do, they actually have to sense the glucose, the sugar in the blood, and proportionally to this to secrete insulin. So this is electron microscopy picture of beta cell. So this is the cytoplasm, it's part of the cell, and what you can see here, these are secretory granules, which are packed, this black is insulin crystals. And they are surrounded by subunits in the cell, which are so-called the powerhouses of the cell, the energy-producing subunits. They are called mitochondria, have awfully difficult name. And the reason how they actually came to exist in the cell is fantastic, fascinating story. They were alpha bacteria. They by some point were engulfed with us in the cell, so they were taken and they begin symbiosis, which nowadays will reflect physiology and health of the cell. These mitochondria, these rugged kind of structures generate energy. And when the energy is generated through them, this is a security, safety lock for the cell to exist as a normal cell. So why they are around the secretory granules of insulin? Because secretory granules need energy to discharge the insulin in the lumen of blood vessels. So now there is one schematic which shall tell you that there is no better monitor, sensor of sugar in blood than beta cell. How does it work? The principle is very simple. So, how beta cell senses how much there is a sugar and how much insulin it has to secrete? It takes the sugar fully from the blood and completely sends to this mitochondria. And in mitochondria, there are two factors to generate energy, sugar plus oxygen. And energy in the cell must be generated in the way to be recognized. This is called ATP. But now, how much ATP you have generated from mitochondria depends on sugar. And then ATP signals these channels, triggers entry of calcium, and calcium discharges insulin from secretory granules. This means it's fantastically linked. So much insulin depends on how much sugar. So there is no better monitor and better sensor designed ever of a beta cell. But what can go wrong? So here you can compare again the island collection of beta cells in normal, non-diabetic patient, and here in diabetes. What can you appreciate is in diabetes there are much less beta cells, right? And we have stained them with insulin to find out where they are, why they are less. So the person actually who attracted me so much with the power of his research to come over here, to leave my research at UCLA in London and to come, is Dr. Peter Butler, and he found that he analyzed the cells and he found that they are massively dying by this self-suicide called apoptosis. So why they are dying? What's happening with these cells in diabetes? Why we are losing beta cells, which are insulin-producing cells? So he found, and I have forgotten to show you something here, he found that together with insulin, there is secretion of one very enigmatic protein in ratio 1:10. And guess what? This protein is present also in the brain, and it can cause neurodegenerative diseases like Alzheimer's disease. And now he analyzed this protein and he found how it is composed, what is the structure, but the most fascinating finding was that this protein with this structure is conserved only in 3 species. Humans, monkeys, and cats. And guess what? These are the species that cats spontaneously develop diabetes. So not in rodents. So now what he decided, let's prove that this protein can be causative for diabetes. He has made use of genetic engineering and expressed the protein specifically in these insulin-producing beta cells in mice or rats. And what happened, they developed diabetes which resembled many features of diabetes in humans. So this proved that there is causal connection between this protein, which is still not known what is actually the basic function, but we recognized in its potential of toxicity. So what is this protein doing when it's toxic or not? So when proteins are just versions of the genes, so they are extended version of the gene and they are bearing function. So protein is a function. Protein has a role in every cell to exert function. For some protein is a key to fold and unfold. So it's very dynamic. in order to exert function. However, with the aging, the processes that have to control this protein decline, so with the advancing age. And it happens that the protein can form association piles, which we call oligomers, and they are very toxic. And the reason why they are very toxic, they can interact with the membranes, and such membranes are our powerhouses where the energy is generated in beta cells, mitochondria. So the first step, what we decided to do is to take beta cells, put them in a petri dish, have controls, and have the toxin, and compare the mitochondria, whether we can identify any changes in these subunits that generate energy in the cell. So we have done this, and here you can see we have stained these mitochondria, these powerhouses of energy generating with special dye. These are controls, and these are cells that have the toxin. Morphology is completely different. Here, mitochondria are networking, they're socializing, they're interconnected. They are part of one big element, no? It's a trait. They are connected, woven. Here, they are fragmented, they are more isolated, they are congested, they're squeezed and almost like anxious. And this is here the nucleus of the cell, so surrounding the nucleus. So we said, but this is in petri dish. Is this the case really in vivo, in tissue from humans? So we decided to stain these mitochondria in humans. Here are mitochondria in beta cells in non-diabetic patients, and here in type 2 diabetes. Here mitochondria are tightly woven and dense, but here are depleted. At least the density, you can see that it is reduced. So it means they are affected even during diabetes, where we have more factors, in the same way as when we take beta cells and intoxicate them with this called amyloid protein. Okay. However, now mitochondria generate energy, right? Remember that sugar plus oxygen, generation of energy. Energy generated by mitochondria is proportional to sugar and will be proportional to insulin secretion. In mitochondria, all metabolites almost are formed. So now, to prove that I'm right, that something is going wrong with mitochondria, I have to check the metabolite composition. So we did so. It's complicated analysis, but what we found that some subset of metabolites were changed. And now we decided, let's analyze it. What has been changed? What this toxic protein has induced. So what we found was that first, the cells were oxidatively stressed, and maybe you have heard this in the reading journals. From oxidative stress, for example, we take a lot of antioxidants to defend our cells from the oxidative stress, so these cells were stressed. Then also mitochondria were not doing their job well, that is what we found. But we found the third thing, which was the most fascinating: beta cells, in due course of adaptation to the toxin, changed the way how they produce energy. And they did in a very particular way, which was very similar to cancer. They produced energy independent of oxygen. They decided to produce it independent of oxygen, which is called Warburg effect. Okay, are we right? We have to prove that. When you generate energy like cancer, you are producing a lot of milk acid, which we call lactate. So we measured the milk acid in the isolated islets from our animals that express specifically the toxic protein, and found 40-fold increase in this milk acid. And here is again very busy scheme, but I will just simply explain what happened. So beta cells, when they are injured by the toxin, what happened, instead of putting the sugar in mitochondria, they divert it. They started generating energy by process they actually cancer cell use, which is called the Warburg effect. And this is key for their loss of function, because now you can see sugar is not coupled to mitochondria and to insulin secretion. So that's why even when they are residual, where they are present in the pancreas, they are not secreting insulin. So we found a way how. So basically by mimicking the way in which the cancer cells generate energy. And this is because they need first to adapt to survive. They are trading off the function for the pure survival. Okay, so now we decided to look who is governing this change. So what is the mechanism that leads to this change in generating energy? Again, very complicated genetic analysis, but it has to tell us through this combination of colors that represent which gene is increased, which is decreased. Blue gene is decreased, red gene is increased. Is to tell us, do we have some pattern? Do we recognize someone footprint of some protein, of some important mechanism? And the most fascinating thing was that we found the footprint of a protein which is called HIF1α. It's a complicated name for complicated acronym. But what is this coming from? It is the same protein that changes the energy generation in cancer. And this protein, believe or not, is very evolutionary conserved. It appeared 550 million years ago, at the point where the atmosphere, the Earth atmosphere, began to change the oxygen. So the oxygen began to fluctuate. And it was very important because at the time, for the animals, for the first life on Earth, basically it secured survival. Because when the oxygen began to go up and down, this protein allowed change to the way energy was generated independently of oxygen. So what is this now protein doing? Why is this important for diabetes, right? So what happens in diabetes, don't take this message wrong, there is no change of oxygen. But when there is injury in the cell, typically in these beta cells, just the fact that mitochondria, the powerhouses, are not working well, has a particular way to stabilize this protein, this evolutionary conserved protein. And then this protein triggers change of the way energy is generated. And this makes these beta cells that they cannot anymore produce insulin in fashion dependent of the sugar. And that is same as in cancer. So they begin to generate energy as in cancer. So now we said, okay, but if this protein has a multiple mechanisms how it is doing, can we identify enzyme that we can use to try to identify in the pancreas that really has changed, and with this tell us that what we have discovered is associated with diabetes. We found this enzyme, it has again complicated name, and we decided to find out whether beta cells that are sick, that are diseased in diabetes are expressing. And now what you can see here is tissue. Green is showing, so it's staining to show the insulin. Blue is showing the nucleus of the beta cells, so the core of the beta cells. And red is showing this enzyme under control of the evolutionary conserved HIF1α. And this is human tissue, type 2 diabetes. So you can see big cells, so huge staining in big cells, which are still there in type 2 diabetes patients, but which are flagging, basically flushing the presence of the enzyme from this evolutionary conserved system. So if we came to the stage that we said, okay, we identified the way how the beta cells lose function in diabetes, and we found the enzyme. And the enzymes have advantage that you can target them. So if we are right, if we inhibit this enzyme, maybe we can promote the survival of beta cells. So we've done this first in Petri dish. And we inhibited the enzyme and then checked metabolite composition, structure of mitochondria, and the survival. Metabolites returned to normal control levels when we inhibited the enzyme. Mitochondria become again networking and woven, as in controls, not anymore fragmented, and there were no any more signs of damage. And cells, this is one special analysis. This here peak shows the number of dying cells. Cells were reduced in dying, indicating that our strategy actually promoted survival of beta cells in the toxic background in type 2 diabetes. So we asked how is possible one enzyme from one stress pathway to inhibit and to secure survival of the cells. So our idea is, during the injury, there was accumulation of a lot of calcium, deleterious calcium, and we tried to analyze it. What we found, so when we have the toxin, there was a lot of calcium compared to the controls. But when we have inhibited the enzyme, we return the calcium back to normal levels, and probably this is the basis how the cells were survived. So these are just showing number of experiments and then levels of calcium control in the toxin or toxin where we have inhibited the enzyme. So, in conclusion, what we have found? We found that one protein that is co-secreted with insulin in order to attend the function, folds and misfolds, but during this process in the aging and decline of our control processes of degradation of this protein becomes misfolded. This is this amyloidogenic protein. And this protein causes changes in the way insulin producing cells generate energy. And this begins to resemble the way cancer generates energy. And the problem is, if you do so in a beta cell, basically you do not have any more chance to produce insulin concordant to the levels of sugar, which is the main function of the beta cell. So we explained the way how it is doing. But why is this relevant for cancer? You might ask yourself, right? So the reason is that this happens in the same time in the pancreas where you have tissue that is prone to development of cancer. This change of the energy production can work and can support development of cancer, in this case pancreatic cancer. The research is going in this direction, we still are not ready. And so something that is most important, it's present in 100% of all cancer, is change of this way of generation of energy, which we call metabolic reprogramming. And we found that this happens in diabetes in injured insulin producing cells. So we identified one enzyme that when targeted, it can reverse diabetic response. We are finding that also partially is recovering the function of beta cells, and currently we are developing this approach for the clinic in the treatment of type 2 diabetes. So now let me cite something which I found. The Bible can basically have vast citation full of metaphors that science may need decades to discover. And I found something really interesting in the book, citing Apostle Paul, in which, interestingly, in the layers of the atmosphere, they explain where there is lower oxygen, not lower oxygen, lower air, exists an evil which is associated with illnesses. This is very metaphoric associated with what we find, that we find basically a general disease principle that is present across cancer and diabetes, and originally it was formed because of less oxygen, but now it seems to empower various diseases. And I can tell you that this becomes now very important as a general disease principle, not only in diabetes and cancer, but also in neurodegeneration. So how wise were the citations and metaphors and myths from the Bible that contained information that it took us very long to understand. But in order not to conclude on these terms, and let's think about the air, let's think about the spirit, and let's think about God. And the opposite of absence of air, of something that potentially generates this general disease principle, is presence of air. So let me cite something which I also found about the feeling of presence of air and spirit. It says, "I'm never alone wherever I am. The air itself supplies me with a century of love. When I breathe in, I'm breathing in the laughter, tears, victories, passions, thoughts, memories, existence, joys, moments, and the hues of the sunlight on many tones of skin. I am breathing in the same air that was exhaled by many before me, the air that bore them life. And so how can I ever say that I am alone?" So with this, I would like to thank you, to thank the people who helped me to execute this research. We are a little group with one technician and one postdoc. And just to say that throughout my work, I felt like, and I will cite again Nikos Kazantzakis, like almond tree asked to speak of God through the work. And when the almond tree was asked to speak of God, "Friends, speak to me of God," and the almond tree blossomed. I felt that at some point my work blossomed because speaking of God. So thank you very much, and happy class.
Can I ask a question?
Yes, please.
Okay, I get actually, I'm suffering from diabetes too, so,
Yes.
and I'm keeping it under control with metformin.
Yes.
But is there a way to reverse it?
So, well, there is, yes, initially, before you are on insulin-dependent therapy, there is always way to reverse it. We hope very much that this approach will theoretically reverse it, because it has to preserve the residual beta cells that you have and also recover their function. So that is the main idea of this work. So
How long before?
15 years.
Too late for me.
Yes. Može.
Da li to samo je za type 2 diabetes ili je i za type 1?
Ovako, u type 2 smo testirali, a u type 1 samo znamo da je isti respons, znači da ist enzim je aktiviran, to znamo, napravili smo preliminarne studije, sad smo dobili od jedne velike kolekcije koja se zove ENPOD na raspolaganju human tissue i sad treba da radimo mnogo više eksperimenta da bi potvrdili. Međutim, postoji mogućnost da je kao, pošto je, ja moram da objasnim još jedanput stvari, težina ovih saznanja je u tomu što to može da bude generalni princip bolesti. Jer u stvari šta je? U našem organizmu postoje tkiva, kao što je, na primjer, kao što je pankreas ili brain, oni nisu dizajnirani da odgovore regeneraciji. Oni nisu dizajnirani za regeneraciju. I problem je kad su oštećeni, na primjer, ovim amiloidnom proteinom, onda ta regeneracija, taj program počne. Međutim, pošto mi zovemo to sustained injury, znači taj problem, to oštećenje traje dugo, ne može da se prekine, nije akutno, onda ultimativno te ćelije se gube i uništavaju. Dok, na primjer, u akut kidney injury, to je druga situacija, pošto je stres akutni, isti ovaj sistem, znači sličan sistem je principijalno počinje kao regeneracija, međutim je uspešan, tako da, na primjer, tamo ćelije mogu da se regenerišu. Tako da u cijelom organizmu možemo da razlikujemo kao modele ćelije koje su spremne, koje mogu da izvrše regeneraciju i ćelije kao, na primjer, u pankreasu ili u mozgu kod neurodegenerativnih oboljenja, gdje oni stanu u tom procesu jer nisu dizajnirane da se regenerišu i zato što samo oštećenje je hronično. Znači ne je akutno kao, na primjer, u kidney, u bubregu. Hvala. Yes.
Ja sam dijabetičar 40 godina i veoma sam oduševljena vašim predavanjem i razumela sam kompletno sve. A meni je lakše da kažem na srpskom, jer sam ja u procesu da sam sad postala neki fenomen u dijabetisu, da ja mogu da ga regulišem preko dana i recimo legnem sa 62 ili ispod 100, a ustanem sa 300. Znači, ja sa insulinom to ne mogu da regulišem. Pošto sam počela da se bavim, inače sam mikrobiolog i hemičar, počela sam da razmišljam da li pH hrana ili uopšte krv utiče na razvoj ili stop dijabetisa, pošto vi ste sad spomenuli kiseonik. A kiseonik je jako bitan u celom ovom procesu, jer rak ćelija ne živi bez kiseonika.
Tačno, apsolutno. Mislim, treba je kiseonik, međutim ona proizvodi energiju i nezavisno od kiseonika.
Sada, da li postoji hiperbazična komora ekstra lečenja sa dodatkom kiseonika? Jer kad ja merim šećer, ja uvek imam oko 95, a normalno mi treba da bude 90, recimo. Jer znači u mojim ćelijama nema dovoljno kiseonika.
Ja vas razumijem, to bi bilo jako interesantna ideja. Nije mi poznato da li takav pristup postoji u smislu dijabetesa, ali bi bilo jako interesantno zato što, na primjer, isto postoji jedno saznanje da ljudi koji žive na većim nadmorskim visinama gdje je razređen kiseonik, da u stvari oni imaju određenu veću sklonost ka razvoju dijabetesa. Tako da u ovom slučaju što sam ja vama prezentovala, znači to je jedan mehanizam koji je evolucijom postao na bazi prisustva ili odsustva kiseonika, međutim koji sada ćelija koristi da bi se adaptirala, međutim kad je hroničan stres ćelija odumire. Sad to je jako interesantna ideja, međutim nije mi poznato da li je testirano. Da li kiseonik bi imao utjecaj. Logično je da bi imao, a ja ću vam reći još jedan podatak, zato što ovaj amiloid o kojem sam pričala, taj je toksičan u ćeliji, međutim van ćelije on formira kao depozite, mi kažemo amyloid fibers. I oni mogu teoretski da izoluju ćeliju od krvnih sudova, što znači da manje kiselnika dođe u ćeliju i to može da bude isto jedan od problema, što kažete da taj manjak kiselnika u stvari proizvodi tu reakciju. Znači, teoretski, jako je opravdano, postoji logika u tome, ali ne znam da li su je testirali.
Da li vam je poznato da li dijabetičari koji su tako dugo treba da se izlože barijatričnoj komori?
Nije mi poznato, ja to bi morala da pitam.
Inače u Beogradu je endokrinolog koji mene vodi Lalić, svetski poznati čovek i tako dalje. I onda mi smo ovako u nekom razgovoru, on me prati kao specijaliste za šećer, mislim, ja možda od 100, ja sam jedini koji takav reaguje. Jer ja nemam prave simptome šećera, a imam visok
to je jako interesantno. A znate što, ja bih samo rekla da vi morate definitivno ne se obratiti, ja bih vas uputila na lekara kod koga radim, u stvari koji je direktor instituta i koji je jedan od vrhunskih specijalista za tretman dijabetesa kojega bi ova posebna situacija interesovala i on bi znao najbolje možda da odgovori vama.
A ja ću vam isto dati informacije, recimo za ovo što ste vi sad pričali, to uopšte nije novo, to je još u početku 1923. dobio Nobelovu nagradu, jedan Nemac.
Otto Warburg, to je u vezi kancera,
Kancera i kiselnika, mislim.
Apsolutno, da.
Ja sam počela njegove studije malo više.
Zato se zove Warburg efekt, da, tačno.
Hvala vam velika, ja bih voljela da dobijem kontakt s vama.
U svakog, bit će meni drago.
Ali jako sam zadovoljna sa predavanjem. Prvo što sam naučila, ono što sam znala, ja sam sad zaoblila.
Hvala.
Znači, ono što ja sad proučavam, je tačno što mi dali da je to tačno. Baš drago mi je što me Blačko pozvao i...
Hvala vam puno. Hvala.
Lena ima.
I don't know what you were talking about because I don't understand the language you're speaking. I have a question. Do you collaborate with some laboratory which is producing, and right now they doing a lot of progress in epigenetics?
Epigenetics. I'm not working on epigenetics personally. I had a collaborator, who worked at my institute, now moved to City of Hope. She is working on epigenetics, where the epigenetics are changes basically in proteins, for example, like histones that are enveloping the DNA, and this changing independently of genes can cause a lot of responses in the cell. Epigenetics plays a huge role, and I can refer you to the literature of my colleague, but also, there are many people working in the field on epigenetics that plays a role in diabetes.
Yeah, it's not. I'm not interested in theoretical things. I just meet a lot of people who is struggling from the cancer, and there is already a particular medicine which is used for people, and they just getting all the statistics from the people who is already survival of the cancer. And your description right now is a perfect description, just because the doctor is more concentrating on the result, he cannot explain, and he doesn't bother to explaining these people how you in detail explaining how it's happening. And the people, when they already in situation, they just go and take, and they offering these things. And
the ultimate kind of approaches, therapeutic approaches, which do not explain exactly the rationale. Yes. So first, regarding the question of epigenetics, plays both role in all the diseases, as I say, but from the aspect of cancer and how it is approached, I mentioned, for example, my previous supervisor from London, UCLA, he established biotech that works on genetic sequencing of the tumor tissue and identifying the unique genetic profile. Then two centers, one from Ireland, one from Italy, are taking a look at as experts, and they're deciding on the best possible concordant therapy in this particular case. So each case is like a subject of so-called precision therapy. So here the rationale is that you cannot put everything in one pot, since they are very specific, but this idea is more evolutionary that actually when you know, it's like when you zoom the picture, when you too much zoom painting, you don't understand what the painting is telling you. But when you little bit take the stance from the painting and the perspective, what we recognize now, and we have many methods, collaboration, for example, at UCLA, that we do comparison with Alzheimer's disease, that actually there is this general disease principle. You know, it might sound like from many centuries ago, but it is the one which just, it's different in different tissues, so that it has some differences in different types of tissue and models. So they're opposing ways. I agree, when you know the rationale, what's happening, it's much easier also for the patient, right, to understand the therapy, what is getting. I agree with you.
Did you familiar with it was a really theory teaching from the 80s from the Soviet Union, and they called the Torsian fields. Torsian fields. It was in an academy of the science, and it was like a big group of the scientists who is studying, and they saying they go into the, like, they combine quantum physics together with the
biological stuff, yeah.
Yes.
I'm not familiar, but I would definitely look at. It would be interesting. I think that the future relies in science on interdisciplinary approach. So we cannot see everything. Not only that we have to take perspective to understand the painting on the wall, but we have to take different perspective: quantum physics, biology, chemistry. I don't know, and that is definitely now necessity, not anymore just the luxury of thinking whether you will choose this or that option. But in order to have these profound insights into scientific findings, you need exactly that. You need interdisciplinary. So I will look, and they're called the Dorsian fields.
I will
if you send me the link.
And I would be really interested in if you have this like presentation.
I would love to contact
somewhere. With your name, with your work, because I would contact you with this laboratory with the doctor.
Definitely, I would love
because
to.
It's really interesting in detail, goes really deeply in a level explaining the things.
I would love to, and thank you so much for that.
So we talked about therapy once you get diabetes, but from this research, what we can do to prevent diabetes, because our lifestyles, as you said, probably lack of oxygen, the air is so polluted, light pollution also. All these lights, they have very high blue spectrum light, but since 2004 I got diagnosed with melanoma, so it changed my perspective on life, and I tried to find the best. And after doing all kinds of nutritional approaches, the thing that I recognize is most helpful life of living, it's following the Orthodox tradition.
Yeah, I agree.
With fasting, with trying to avoid eating food for pleasure, not to. If you follow the Orthodox tradition, you're safe.
No, I agree. And there are scientific findings that completely confirm this, and especially the fasting. So now
intermittent fasting.
Intermittent fasting. And there is always question how much, but basically it's based on your daily intake. So if you can reduce even at some days so during the days intermittently, 80% of this daily intake, that you are doing well in terms to help yourself.
So for example, if you follow the Orthodox rules, you fast on Wednesday and Friday.
Exactly. That is what I said, that interestingly the Bible, the tradition, it has such a wise ways of treating life and health, and that it takes science, unfortunately, very long to come. Maybe it's coming with details, but it's coming to the same findings that are incorporated in the tradition. I agree with you. And the fasting, now there are spectacular scientific findings that were recently published to demonstrate what is the mechanism of recovery. I agree with you completely. Please.
But what about, because I've been reading a lot about inflammation and all that stuff, and beans and seeds, they produce inflammation, which is basically the number thing to all of the diseases.
I would tell you just one thing. So if you, we call this kind of monothematic food. If you eat just broccoli, I don't believe that you will help yourself, or you eat some just seed or whatever. The nature of the diverse, when you eat diverse food, and important is to be fresh and unprocessed, right? And not to have some critical addition of pesticides or artificial substances. Then the idea is that the food, also the toxins in the food can counteract itself. So I would actually advise you against having the idea of concentrating on only one type. Beans, I don't know, I am not aware that beans can cause inflammation, but beans are all the beans family, so-called Fagaceae family, is very well known for presence of proteins, and these are absolutely key. I remember when I worked in Vienna, I worked at the Institute for Oncologic Toxicology. They were together with garlic and white cabbage, number 3, I would say, in anti-cancer, in cancer prevention denominated food. So my idea will be, rather than focusing on and sticking to some types of food, advising on diversity, because when you have diversity, the food is designed also when there are negative parts like toxins, poisonous part of the food, they counteract each other. So it's, of course, moderacy in feeding, it's important, and as I said, fresh, unprocessed food. But you will always find parts, for example, soya, it's fantastic because it has the so-called weak estrogens. But it's fantastic in Japan, where the people start, young ladies start to consume soya very early, so it can really work. So it can kind of counteract the estrogens throughout a very long period, and then works as a cancer prevention of breast cancer. But in West societies, you know, soya is just up a little bit here and there, and I would not believe it might be. It's advisable to be part of your food, but in the whole diversity of your nutrition. I would not focus on one. And another thing which I may mention to you, because I was part of these studies in Vienna, is that it's important to, now we live in kind of, especially here in California, you're exposed to all sorts of food. But it's interesting that when you originate from various parts of the world and you are locally more exposed, it is more advisable to use the food which is characteristic for the region rather than to expose to more exotic type of food, because apparently it takes many years, and this food behavior and adaptation to the types of food took many generations in order to be maximal for positive for your organism. But here, of course, it's very difficult to apply because we have kind of we are exposed to very different types of food. But that is what I actually learned at the times when I studied. Important as bearing message diversity in food, not only one.
Balance. It is really talking about balance.
Yes, yes.
Ja nisam lekar, sam pedagog, pa mogu odgovoran da kažem da vam čestitam na izvanrednom predavanju. To mi je struka i više o tome govori pažnja sa kojom su vas slušalci cijelo vrijeme slušali. Ja imam jedno kratko pitanje. Znam šta je dijabetes, znam šta je normalni šećer u krvi. Ja mislim da je poznato u medicini i nešto treće. Ljudi koji imaju nizak nivo šećera u krvi. Ja sam poznao čovjeka kome, kada je konstatovan u prošlosti šećer samo preko urina, konstatovan je dijabetes. Čak ni u kafu nije smeo kašću i 20 godina tako živio. Razbolao se i morao je na operaciju, ide, i kad su ga lekari pitali, događalo se u Njemačkoj, kad su ga lekari pitali šta je sve boleo, kaže ja 20 godina sam od dijabetesa. Dobro, vidjet ćemo, kad su izvadili krv, čoveče, pa vi nemate nikakav dijabetes. On je zadnjih 10 godina nemilosrdno tamanio šećer koji mu nije škodio. Ustanovljeno je da ima gubitak šećera preko urina. To je neka deformacija, ne znam šta je, ja nisam lekar, ali hoću da kažem da je i takav slučaj. Mislim da postoji u medicini mišljenja o ljudima koji imaju upravo obrnuto nešto diabetes.
Apsolutno, i postoji i terapija koja je bazirana baš na tome korišćenju, znači, tih transportera šećera u urini i koji u stvari šta rade, znači oni namaljuju, jer kad šećer dođe i kad se formira konačna urina, znači treba da se izvrši resorpcija šećera u krvi. Međutim, sa tim lekovima može da se spreči tako da se više izvlači iz organizma, na koji način se namaljuje šećer u krvi. Znači to što je vaš prijatelj imao kao jedan, da kažemo, deficit ili deformaciju, eto to je sada se koristi u terapiji. Mislim, jedan je terapeutski pristup.
Hvala.
Da, može. Yes.
You know, I think about the GCE math, the search about that, the GCE math.
What is GC meta? I just
GCE math, MAF, you know, search about cancer or stuff like that.
Yes.
You know, any idea about the treatment, about the...
Okay. What about, I have not understood you. You are asking me about possible treatment of diabetes?
Yeah, what they call the GC math.
Okay.
Yeah, well, they talk about, a lot of doctors talk about this treatment.
GC met.
Yeah, you never heard?
No.
MEF.
No. What is it?
Yeah. Can you spell it?
GCMAF.
What does the AC stand for?
What is this staying for? Do you know?
Oh, no. I don't know, but
We can Google.
Yes, I don't know what you mean. Is the, what? Can you just explain me what is? I mean, what is the approach? Maybe it will ring the bell, so I will immediately tell you. What is this about? It's a type of treatment of diabetes.
For cancer.
For cancer, GC-MET.
Yes.
I don't know.
Yeah.
I just looked it up.
Yes.
It says how
Google.
GCMAF cures cancer.
I don't. What is GC-MET? I don't know. Sorry about that. Alternative therapy. So I am sorry, I have not heard. And what kind of alternative therapy is this? So do someone has more information? What kind of alternative? GCMAF. It's apparently alternative therapy, right? It's alternative therapy?
Well, so you can just focus on your own research.
Yeah.
Okay. No. No. Okay. If it is the immune system, and if it not, then I can give you. I don't know the GCMAF, but I can tell you how immune system. If it refers to the immune system, there is something which came last year's. As a novel approach, and I will tell you how. So I started also talking to one of the people in the here present. So what happens? Cancer arises from our own cells, and basically our immune system is designed not to recognize something that is yourself. So this is through the generation of our immune system. Immune system learns everything. If immune system makes mistake and recognizes something which is self as bacteria or virus or tumor, these cells, these immune cells will be discarded. So it's a very strong rule. So everything what is mine, it has to be respected, preserved, so I cannot attack myself. Cancer cells arise from our own cells and cannot be recognized by the immune system. So we say they evade the immune surveillance, right? Because immune surveillance, our immunocompetent cells, can detect cells infected with viruses, detect the cells with bacteria, but not something that arises from itself. So if this, I don't know what is GCMAF, but it rings the bell about a new therapy, which is called immunocheckpoint therapy. So what people have found is, even in the worst types, more advanced types of cancer, if you use special drugs to activate the immune, so they're called immunocheckpoints, then immune system begins to recognize tumor cells and begins to attack them and can eliminate them over a very short period. So people with very advanced cancer actually recover within relatively quick period. But everything comes with a price. So the price is when you have such a boost on the immune system that these kind of forbidden ways of recognition of selves are activated. Self, I mean self cells. And when you activate them, then tumor cells become visible for the immune system. Then this patient become prone to development of autoimmune diseases. So it comes, and they can also develop type 1 as type 1 diabetes. So it comes with a price. So that is a known therapy. It has been established in melanoma. It's very efficient now recently. So this immunotargeting approach. There are a couple of other cancers, and it's spectacular in the way that very advanced, so almost terminal cancer, if they respond to this therapy, they can be recovered. So this is called immunocheckpoint therapy. Yeah, please.
What about hypoglycemia?
Hypoglycemia. So
Low blood sugar.
Yes. So what I understood from the expert I work with, and I personally, I never worked with patients, and I started on the basic research 2 years ago. But from Dr. Butler, what I understood, it's more dangerous hypoglycemia if it is induced than hyperglycemia. That is kind of can be sustained. Yes. So hypoglycemia, the incidence of uncontrolled reduction of the normal blood sugar can be actually fatal. So that can be really controlled. So that's really important. And I remember discussing one case that luckily I was good hearing. So I remember the danger of the hypoglycemia as such. So it can be really dangerous. Yeah.
Made it this far? It's all good. I trust God. He's got me. Father Blasco, anointed among you nations.
Okay, one big round of applause. It is absolutely amazing. Can I have your attention, please? How thorough, how detailed, how professional Dr. Slava is. Thank you so much, and I pray that God multiplies your talents and brings the benefit to the mankind. You know, I mean, these are the people who work and help the whole mankind, and we all, you know, really express our gratitude to you. And this lecture is a great gift to our church and our community. And the greatest gift is you as a person, and embracing our church and our community together with your lovely daughter. And I cannot express enough my gratitude on behalf of all of us. And I hope that, God willing, we will hear you again, here, you know, to talk to us and to share the wealth of your knowledge. Thank you, Dr. Slava. And also today she brought her friend. I didn't have time to introduce in the church. Your name again?
Jelena.
Jelena, dobro nam došli.
Hvala.
I da nam dođete ponovo.
Hvala. Doviđenja.